Hello again. You have my voice? Okay, if you say anything, Mr. Haidar. I told you if you want it, please tell me. Hear me? Okay. Is my voice clear? Other students? Okay. The sound is cut off. I think because it's your internet problem, you can refresh Fatima. Refresh, please. Okay. I just want to compare prokaryotes and eukaryotes transcription. So now we want to talk about transcription. Transcription. Okay. Transcription. As you know, we have DNA. After that, after the process of transcription, we have mRNA or RNA. After mRNA, we have proteins. Okay, so at first, DNA. Transcript to RNA, and after that, by the process of translation, we have what? We have protein. Okay, what's the difference of prokaryote and eukaryote transcription? At first, has the promoter, but eukaryote promoter and enhancers. Prokaryote promoter, eukaryotes have enhancer. What I mean, is the region in where the RNA polymer can bind. So, motor in the gene which RNA polymer can find it, transcription start. Okay, but in we have promoter and RNA polymerase RNA. We have RNA polymerase in prokaryotes, but we have 3 RNA polymerase eukaryotes. So, just 1 RNA polymerase in prokaryote, and we have 3 different RNA polymerase in eukaryotes. We have the factor rho. We have the factor rho in prokaryotes, but we don't have rho in eukaryotes. What is rho? Rho is the protein which play a role in termination of transcription. So rho is a protein we play a role in of transcription. Have rho in prokaryotes, have rho in prokaryote in processing the replication is much processed. Hear me? Ale? My voice is good? Okay, let me refresh. Can you hear me? My voice is good? Okay. I was talking about the different parts from transcription of prokaryote and eukaryotes. Okay, I told you we have promoter in prokaryote and we have promoter plus enhancer in eukaryote. I told you where is the promoter? Promoter is where RNA polymerase find the region and transcription start. So the promoter is the region which RNA polymerase find it and start the transcription. We have only one polymerase in prokaryote, but we have 3 different RNA polymerase in eukaryote. We have rho. Rho is the factor which play role in termination of transcription, but we don't have rho in eukaryotes. In prokaryotes, not as much processing, but the transcription is processing in eukaryote. What does it mean? It means we have so many processes in eukaryotes. For example, splicing, for example, capping, for example, polyadenylation. I'm sure you studied about them, but we don't have them in prokaryotes. Which one is slower? Which one is slower? As you can see here, the transcription of prokaryotes are slower than eukaryotes are slower than prokaryotes. Okay, so thank you. This is the very short comparison with eukaryotes. Let's talk about the RNA polymerase of prokaryotes. RNA polymerase of prokaryotes. As I told you, we have only one RNA polymerase in prokaryotes. So we have only one RNA polymerase in prokaryote. We have different parts. Look at this picture. We have alpha, 2 alpha, alpha 2, beta, beta prime, omega, and sigma. So we have different parts in RNA polymerase in prokaryotes. Alpha 2, beta, beta prime, omega, and sigma. Okay, without sigma, we call this enzyme core enzyme. But after sigma add to this, we call it holoenzyme. Can you tell me what does core mean in Arabic? Can you tell me please what does mean in Arabic? Okay, so I think we can tell it markaz. I don't know what you tell it. So core RNA polymerase or the middle of the enzyme made up of alpha 2 beta omega. After we have C, we call it holoenzyme. So repeat again. We have alpha 2 alpha, alpha 2 beta, beta prime, omega, and what? Sigma. Thank you. And sigma. So this is the structure of RNA polymerase in prokaryotes. What does sigma factor do? Sigma factor just find the promoter and start the transcription. So what is the function of sigma factor? Sigma factor can find the promoter and start the transcription. What will happen if we don't have sigma factor? The transcription takes so many times to start, or it can be it can never start. Okay, so sigma have the very important role in start the transcription. It can find the promoter. Understand? Did you learn it in your bachelor degree? Did you had it in your bachelor degree? Okay, thank you. I want to talk about the promoter of prokaryotes. The promoter of prokaryotes. We have two boxes, as you know, -10 and -35. We call -10 Tata box, Tata box. Or do you know another name of Tata box? Do you know another name of Tata box? We call it Pribno box too. Pripno is the researcher which find it. His name was Pripno, so we call it Pripno box 2. So we have two different regions. One is -10 and one is -35. These regions are very conserved. Very conserved. What does it mean, the regions are very conserved? It means that during the evolution, there was no mutation in this region. What will happen if we have mutation in this region? We have problem in transcription. So if we have mutation in this region, the transcription doesn't start. Okay, so we don't have gene expression. We have very conserved region of -3 and -35, and also the distance between these two are important too. For example, the distance should be 17 plus and -1. 17 plus -1. What does it mean? It means that if the distance between -10 and -35 is about 70 nucleotide, the transcription starts. But if, for example, we have 19 nucleotide here or 20 nucleotide here, the transcription can't start. Okay, so the distance between data box and -35 region is very important. It should be about 17 plus -1 nucleotides. And from the middle of -35 to -10 is about 23 plus -1 nucleotides. So from the middle of -35 to -10, it should be around 23 plus -1 nucleotides. So the distance is very important. Understand up to now? Do you want me to repeat? Okay. I am talking about the promoter structure. I'm talking about promoter structure. Okay. In the structure of prokaryotes, have two very important regions. The first one is tata box. We call it -10 region. This is the nucleotides T box. We call it -10, and we have -35 region. -35 region. Okay. And I told you this is very conserved. What does it mean very conserved? It means if we have some mutation here, the replication can't start. So it should be very conserved. We shouldn't have any mutation here. Also, I told you that the distance between -10 and -35 is important too. So we should have 17 plus -1 nucleotides here. If, for example, we have 18 is okay, we have 18 is okay. But if, for example, we have 20 nucleotides between, we have some problem. Understand? The distance is 7 plus minus 1 nucleotides. Yes? Okay. From the middle of this region, it should be about 23 plus minus nucleotide. What does it mean? It means middle of this to middle of that should be about 2 or, for example, 24 nucleotides. If there is more nucleotides less than these nucleotides, we have some problems. Okay, can you please tell me where the transcription starts? Where the transcription starts? You can see it in the picture too. Where transcription starts? Plus one site. Yes, thank you. So the transcription starts from plus one site. Here we have one purine nucleotides. Here we have one purine nucleotides. Can you please tell me which nucleotides are purine? Which nucleotides are purine? For example, adenine and what? Guanine. Do you know purine and pyrimidine? Okay. No, Fatima. Are you kidding me? So we have Ag here, purine nucleotides. It should be A or G here. Okay. Look at this picture. I told you we have two different important regions in the promoter of prokaryotes. One is -10, we call it Tata box, and -35. Okay, and I told you transcription starts from +1 region. As I told you, RNA polymerase attached to promoter, and transcription starts from +1 region. Okay, we have mRNA here. Look at this picture. Left side of Tata box, we call it upstream. Left side of Tata box, +1, we call it upstream. Right, look at here. Right side of the +1, we call it downstream. So left side we call it upstream. Right side we call it downstream. So can you tell me where is the promoter of prokaryotes? Is it upstream or downstream? downstream, upstream, upstream. Look at here, the promoter is here. I told you here is upstream, yes? And here is downstream. Look, left side is upstream, you can see it in the picture, yes? And right side is downstream. Understand? Okay. Where the promoter bind? Where the promoter bind? Look at this picture. The pink one is RNA polymerase. You can see it in the picture. The pink one is RNA polymerase. It can attach from minus 50 to plus 20. Plus 20. So, how many nucleotides can it cover? Look at me. Please look at the picture. From minus 50 to plus 20. How many nucleotides? 70. So, what is beneficial? What is beneficial? When the DNA become single strand, exonuclease may degrade the DNA. So, the RNA polymerase attachments can protect the DNA from exonuclease 2. Can protect from exonuclease. Okay? So, let's talk about initiation of transcription. So, in transcription, we have 3 different parts. One is initiation. One is initiation. Second is elongation. Third is termination. So, at first, we want to talk about initiation. Initiation itself has 3 parts. One is seeking for promoter. Second is making transcription bubble. Transcription bubble. The third one is primer synthesize. So, in initiation of prokaryote transcription, we have 3 different parts. First, seeking for promoter. Second, transcription bubble, making transcription bubble. Third, primer synthesize. Okay? Look at here. Look at this picture. Holoenzyme attach to DNA and find the promoter. Do you remember which particle finds the promoter? Do you remember what I said? Which particle finds the promoter? Can you tell me? Which part can find the promoter? I told you, look at this picture. I told you we have alpha to beta, beta prime, omega, and sigma. Which particle can find the promoter? Sigma. Remember? Yes, thank you, Fatima. So, thank you, Amine. So, the first step of seeking for promoter, sigma factor, find the promoter, attached to it, and after that, the transcription bubble starts. Look at this picture. So, holoenzyme attached to DNA, find sigma factor, find the promoter. After that, the DNA should separate and we should have bubble of transcription. How? Listen to me. We have no factors. RNA polymerase change its structure and open the DNA by itself. So, RNA polymerase change its structure and change its isomerization. So, by changing its structure, it can open DNA. We call it what? We call it transcription bubble. So, after that, the third step of primer synthesis. We should have 7 to 12 nucleotides. For initiation of transcription. But if we have less than 7 to 12 nucleotides, the transcription doesn't start, so we call it abortive initiation. I'll repeat more. Listen to me. In primer synthesis, the RNA polymerase synthesizes 7 to 12 nucleotides for primer, as a primer. If there is less than 7 to 12 nucleotides, for example, 5 nucleotides, what will happen? The transcription doesn't start. We call it abortive initiation. So the length of the primer is so important. How long should the primer be? 7 to 12 nucleotides. Now, I told you if it is shorter than 7 to 12, for example, 5, the initiation doesn't start. The transcription doesn't start. So it should be 7 to 12. Okay? Thank you. After that, sigma factor release or sigma factor disassociated, sigma factor go away, and we have one factor called NOS A. And after that, elongation starts. So, I repeat the whole process one more. Listen to me. We have promoter, DNA promoter, and we have holoenzymes. Okay? At first, holoenzymes find the promoter with the sigma factor find the promoter. After that, RNA polymerase change its structure, and by changing its structure, it can open DNA. When the DNA is open, we call it transcription bubble. After that, we should synthesize the primer. How tall should the primer be? 7 to 12. What happens if the primer is shorter? If the primer is shorter, for example, 5, 6, 4, the transcription doesn't start. We call it abortive initiation. After that, sigma factor release, sigma factor go away. We have the factor name NOS A factor, NOS A comp, and elongation start. Okay? Do you have any question? Okay, thank you so much. Here, the promoter unit can find the promoter. Sigma factor can find the promoter. Yes? Sigma factor can find the promoter. We call it sigma 17. And after that, in the elongation, sigma go away. What we'll have here? What protein should be here? Can you tell me? We should have NOS A protein. We should have NOS A protein. Okay? Let's talk about the termination of elongation. Listen to me. Listen to me carefully. At the end of the elongation, if, for example, this is DNA and this is RNA, we have one palindromic GC-rich region. Did I told you about the palindromic region? Do you know what is palindromic region? Can anybody tell me what is palindromic region? Anybody can tell me what is palindromic region? No. Is a twofold inverted symmetry. Yes, Roya, thank you. Twofold inverted symmetry. What does it mean? For example, listen to me. If here is, look at here. Is a nucleic acid sequence that read the same number matter from 5' end of the sequence itself or from 5' end, it's complementary. Okay, I told you, I wrote it to you, a twofold inverted symmetry. For example, if here is GC, here we have G, C, G, C. So, what does it mean? It means if you read it from this to this, we have, listen to me, G, C, G, C. And if you read from this part to this part, we have G, C, G, C. We call it twofold inverted symmetry. We call this palindromic region. Understand? So, palindromic region is the region. For example, if you read from this part, GC, GC. If you read from this part, you have GC, GC2. Understand? This is the same. If you read from this part, and if you read from this part, this is the same. Yeah. Please imagine if the RNA transcript from the region which is palindromic, what will the structure of RNA? Would you please imagine? Imagine what will the structure of the RNA which transcript from palindromic region? Because the region is the same, it can be form hairpin structure. Yes, it can form hairpin. So listen to me. If the RNA synthesized from palindromic GC rich region, we have hairpin region. So if the RNA polymerase reach the hairpin region, RNA polymerase pause, stop a little, pause from some seconds, and we have two different factors. We call it GRE1 and GRE2, which come GRE1 and GRE2 come and open the hairpin and let the RNA polymerase to pass. So, I will repeat it again. Listen to me. In the elongation, I told you, at the end of the gene, we have the palindromic GC region. We call it pause region 2. So, at the end of the gene, we have palindromic GC region. We call it pause region 2. What does pause mean in Arabic? Max? You call it max? What do you call it? Pause. Anybody knows? What does pause mean in Arabic? توقف. Yes, thank you. Yes, thank you. توقف موقت. Yes? Okay. Very good, very good. So we call it توقف region. So I repeat again, listen to me. At the end of genes, we have the region, we call it توقف region. We call it ناحیه توقف. Okay, توقف region or pause region. Which is palindromic GC rich. Palindromic GC rich. What will happen if the RNA transcript from palindromic GC rich? Because the region is palindromic, the RNA will form hairpin region. The RNA will form hairpin region. RNA polymerase. Pause here, توقف here, pause here, and can't pass from this region. We have two different primer, GRE1 and GRE2 comes, open the hairpin structure, and let the RNA polymerase pass. Let the RNA polymerase pass. Understand? Anybody? Do you have any questions? Okay, thank you so much. Thank you so much. What is GRE? I told you GRE1 and GREB are two proteins which open the hairpin. Look at here, this is hairpin, okay? Which open the hairpin structure of mRNA, which open the hairpin of RNA structure, and let the RNA polymerase pass. I told you because the hairpin structure, RNA polymerase can't pass. So it max here, it pause here, it توقف here, okay? So GRE1 and GRE2 come and open the hairpin structure and let it go. Okay? Thank you, Fatima. So let's talk about the termination. Let's talk about the termination. I want to talk about the termination. Just the GC region at the end is palindromic. GC region at the end is palindromic. Just at the end, not all of them. Okay, so we have two different termination in prokaryote. We have two different termination in prokaryote. One is row dependent. One is row independent. One is row dependent and one is row independent. Okay, listen to me. I want to start with the first one. Independent, first or second? Let's talk about row independent. Row independent. Listen carefully. At the end of some genes, we have GC palindromic region, GC, palindromic region, and after that we have polyorigin. Polyorigin. Okay, so would you please help me? After the RNA transcript from palindromic region, what is its shape? It can form a hairpin structure. Yes? So, after that, it will form U, 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 U. Yes? How is the strength of U, U, U and O? How is the strength of hydrogen bind? The hydrogen bond of A and U is strong or weak? Adenine and uracil, yes. What is the strength? How about the strength of the bond? No, A and U. How many hydrogen bind? How many hydrogen bind? 2, yes. So if we have 2 hydrogen bond, we have weak bond, yes. If we have CG region, we have strong. But if we have A and U, the strength is weak. Okay? So, listen to me. At the end of some genes, we have palindromic region, which RNA form hairpin, and after that, we have polya, which RNA form UUU, poly-U. So, because the hairpin the hairpin bind to DNA very weak, and UUU attached to DNA weak, because the hydrogen bond is so weak, the RNA separate. I will repeat it again. Listen to me. At the end of some gene, we have GC palindromic region, and after that we have poly A. Okay. If the RNA transcript from GC-rich palindromic region, it will form hairpin structure, and after that it will transcript UUU, because their attachment is very weak. RNA and DNA separate. Understand? Do you remember NASA protein? NASA will help this separation too. So NASA will help the separation too. This was the row independent termination. I want to talk about row dependent. Listen to me. So look at here. In this picture, this is DNA. This is DNA in this picture. This is GC-rich palindromic. So the RNA which transcript from here is hairpin, form hairpin. Look at this picture. And after that, we have poly A, which form UUU RNA. So because their hydrogen bond is very weak, the RNA separate from DNA. Okay? Understand? Thank you. So let's go to row dependent. Row dependent. row dependent. I told you some of genes has palindromic, GC palindromic, and after that poly R. But some of the gene doesn't have this region. So some genes have the GC rich palindromic and poly R after that. But some of the genes doesn't have this region. So how would they separate from the DNA? We have some protein which we call it rho. Rho is helicase. I told you before. What does helicase do? Rho is helicase. Helicase can separate two strands, so it can separate DNA from RNA. It is ATPase too. What does it mean? It means it needs energy for this separation, and it is hexamer. So listen, some of the gene doesn't have GC palindromic region and poly R. So in these genes, rho, which is helicase and ATPase, bind to RNA strand where the region which is C-rich region, C-rich region, C-rich region attached to it, and because it is helicase, it can separate the RNA from DNA. Understand? Do you want me to repeat? Okay, listen to me please. I told you some are row independence. What is row independent? They have GC rich palindromic and after that they have poly R. Okay, they can separate because of their structure, but some of the gene doesn't have this region. So how can the termination happen? They have one proteins, we call it rho. We call it rho. Rho is helicase. So what does helicase do? Helicase can separate the hydrogen bond. Yes? So it can separate DNA from RNA. Rho attach to a region in the RNA, which we call it C-rich region, which we call it cytosine-rich region. Rho attached to RNA, and because it is helicase, it can separate RNA from DNA. And it is ATPase. What does ATPase mean? It means it needs energy of ATP. It needs energy of ATP. By the help of ATP and by the function of helicase, it can separate RNA from DNA. Okay? Anybody has any question? So, this is enough for this session. I will finish now. If you have any question, please ask me. Thank you so much. Have a great day. Have a great day. Thanks for your attention. I simplified the lessons, so don't worry, study the lessons which I taught you. Study all the lessons which I told you. Study the region when I told you, which I told you, they are so important. Okay. The previous and this lecture, I will give you some notes. Okay, I will give you some notes. Okay. Thank you so much. Have a great day.